Thank you so much for that kind introduction and the invitation to speak today. I was sad not to be with you in Cambridge because of the COVID exposure you mentioned before this, even more sad now after hearing John give his introduction. But I'm delighted to be able to join you from my home in Brookline and to share with you some of our work, as well as to hopefully lend some perspective, both clinically and scientifically, for the fantastic talks you're sure you hear later today. These are my disclosures. Most relevant to this talk is the advent of BCMA and GPRC5D CAR T-cell therapies and licensed. So in the last few years, there's been an explosion of outstanding clinical trial results that have led to FDA approvals in the CAR T-cell space. These top four here are for CD19 malignancies, and then more recently for multiple myeloma targeting BCMA. And just to shine some perspective on the really transformative impact that CAR T-cell therapies have had in these relapse refractory, heavily pretreated patients with blood cancers, I want to highlight this paper in New England from 2019 of selinexor that led to its approval for relapse refractory myeloma. 2019 wasn't that long ago, but you could see the approval was based off of data with an overall response rate of 26%, a median progression-free survival of 3.7 months, and overall survival in these patients of 8.6 months. Fast forward to the recent approval of cilta-cel. This data was published in 2021 and treated a very similar patient population. You can appreciate the data really speaks for itself with an overall response rate of 97%. And at 12 months, the PFS rate was still 77% with an overall survival at 12 months of 89%. So this is really game-changing for these patients that have failed most, if not all, FDA-approved therapies before going on to CAR T-cell therapy. However, there's still more work to be done. It's not clear if or how many of these patients will have truly durable responses. And even in the CD19 malignancies, where around 40% of patients do have truly durable and likely cured responses, still there's plenty of room for improvement. So I'm often asked what I think about the field of cell therapy, and as you just heard, I think we've had a great first inning, but it's a 9-inning ballgame. What I'm really the most excited about is to see where the field goes from here. So today, hopefully, I can give you some insight into that, and I'm happy to share with you some of our work as a model, thinking about new targets and logic gating, and then also where the field is going, and a lot of this will come from the talks that you'll hear after mine, but next generation chimeric antigen receptor design, intracellular targets, other cell types beyond T cells, vectors, mRNA as a therapeutic and as a target of therapeutics, and then advances in manufacturing and overall manufacturing platforms. So I'll start talking about GPRC5D as a novel target for the immunotherapy of myeloma, and how we translated that finding into chimeric antigen receptors that have taken to patients with some really outstanding early clinical results, and how we think about possibly pairing two targets in hematologic malignancies. So GPRC5D, we first identified it through publicly available transcriptome data, and just give you a couple of samples here. This first is from the Cancer Cell Line Encyclopedia, where there are 1,000 different cell lines on the x-axis here, and that tall red bar are 30 myeloma samples. And it's quite apparent that GPRC5D mRNA is specifically expressed in cells that are derived from myeloma and plasma cells in general. And here's normal patient data. In the top right, there is myeloma bone marrow, and then all the other bars represent different normal tissue data. And again, the difference in mRNA expression is readily apparent. So this is an orphan 7 transmembrane receptor with no known ligand or biologic function as of yet. And in addition to myeloma, you can see those outliers there in the normal slide. GPRC5D mRNA is expressed on a subset of cells in the hair follicle and hard keratinizing tissue, but we thought that this potential for on-target off-tumor toxicity was likely acceptable, and went forward investigating this target for novel CAR T-cell therapies. So first, that was all, of course, mRNA data. We proved to ourselves that GPRC5D is expressed on the cell surface of myeloma plasma cells. Here it's co-localized with BCMA and CD138, another typical myeloma marker with good cell surface staining. And we investigated over 80 primary samples here to examine the relationship between BCMA expression and GPRC5D expression. And to point out just two things about this, that most of the patients fall in this top right quadrant here with high BCMA and high GPRC5D expression. And then that there really was no relationship between the patient samples that had high BCMA versus high GPRC5D expression. So we were encouraged that these were two potentially orthogonal targets. We then went on to undertake SCFV phage display panning. These are library derived from human B cells. And I won't take you through all of this, but ultimately identified 7 clones that were highly active and specific in terms of binding GPRC5D. These bound different extracellular loops of the GPRC5D transmembrane protein for epitope mapping. And ultimately, we engineered 42 different chimeric antigen receptors through varying the orientation of the variable heavy and variable light chains, or investigating multiple spacers. And for time's sake, I won't go through all of this data, but we employed first in vitro techniques such as proliferation after repeat antigen stimulation and lack of tonic signaling to identify a lead candidate, and then we confirmed in vivo that that lead candidate was in fact superior in terms of anti-tumor efficacy and survival in mice, to want to bring that forward into subsequent studies. And I will show you a model that we developed for antigen escape. So here we've CRISPRed out BCMA from a myeloma cell line that endogenously expresses both BCMA and GPRC5D, as most myeloma cell lines do, and then put in GFP pyropolyluciferase specifically into those knockouts, and injected mice with a pooled myeloma cell product that contained a minority of the BCMA knockout cells. And then here we're imaging specifically the BCMA knockout cells. And we treat these mice either with irrelevantly targeted negative control CD19 CAR or BCMA CAR, and as expected, the BCMA knockout cells continue to grow. And then we tried to rescue them either with a second dose of the BCMA CAR or our GPRC5D lead candidate CAR T cells, MCAR-109. And when we flow the cells from the marrows of these mice at the end of the experiment, what we can see here is for the negative control mice, there are 2 populations of myeloma cells indicated as myelomas. They're high for CD138 on the y-axis. There's a wild-type population, and then a knockout population with a GFP surrogate for the knockout. When we treat with the 2 doses of the BCMA CAR, we can eliminate the wild-type population. So this is a faithful model of antigen escape relapse. And of course, when we treat with our lead GPRC5D CAR, these mice can do well long-term. So this construct is initially translated as a single-center study with academic manufacturing, and that was recently presented at ASH, and I'm happy to share that with you in a slide or two. And it's also open now as a multi-center trial, including at Dana-Farber, and should credit Shamel and Cody for leading the single-center study, and Omar Nadim is the PI at Dana-Farber, is actively enrolling patients. So just to set the stage for those clinical trial results, these are results from BCMA CAR T-cell trials stemming from our prior lab work. And just to highlight, these patients can have enormous disease burden, which really gets rapidly cleared by the CAR T-cells. And as you saw with the cilta-cel data here with this Orva-cel construct on the right, has a 92% response rates, with many patients, the majority of patients, completely clearing their myeloma paraprotein from their blood, which is what's listed on the y-axis here. So the question we asked would then be, how will patients that oftentimes have these deep responses to BCMA CAR T-cell therapy and then go on to relapse, how would they respond to GPRC5D targeted CAR T-cell therapy? And so this trial did enroll those patients, many patients who had failed BCMA therapy. And in terms of safety, this was very well tolerated. There was one patient in the highest dose level that had severe cytokine release syndrome. That same patient had severe neurotoxicity, both resolved. And in terms of on-target off-tumor toxicity, most patients actually did have grade 1 nail changes, which is unique to this CAR product, likely because of the GPRC5D expression there. And some patients had rash or dysgeusia as well. In terms of the hematologic toxicity, this was pretty standard for CAR T-cell trials after the lymphodepletion chemotherapy. So this is patient number 1 who received our lowest dose of 25 million cells, and this patient had these 2 huge lesions outside of the bone marrow. 4 weeks later, this patient's PET scan looked normal. And overall, we 16 patients that we were able to report back a couple months ago at ASH with a 69% overall response rate, 100% response rate at the highest dose level, and 50% of the patients achieving minimal residual disease negativity in their bone marrow. In terms of the patients on the study that had prior BCMA therapy, there was an 80% response rate, and even with prior BCMA CAR T-cell therapy, a 75% response rate in these patients. So we're really encouraged by that, but still wanted to ask the question, instead of treating patients that have relapsed after BCMA CAR T-cell therapy, could we combine BCMA targeting with GPRC5D targeting and prevent antigen escape relapse, or relapse that's mediated by a low antigen reservoir in the first place? And so this is a summary commentary from Stan Rydell's group that accompanied a paper of ours in Blood Cancer Discovery, which I think does a really nice job explaining the current situation. So currently we're treating patients with BCMA monospecific CAR T cells and leaving behind this reservoir of green cells that are BCMA low, and potentially in some patients there are these cells that are BCMA and GPRC5D double low, which go on then to cause relapse. But if we could target these cells with a dual CAR, with a dual targeted CAR approach, we can at the very least eliminate those green cells that were BCMA low. And hopefully in some patients, I showed you data earlier that BCMA and GPRC5D are really independently expressed. Hopefully in some patients, these double BCMA low, GPRC5D low cells may not exist. And the goal, of course, then is we can eliminate every last myeloma cell. Now, there are multiple potential approaches to go on to create a so-called OR-gated CAR, targeted CAR. There's a CAR pool approach where 2 CAR T-cell products can be made in parallel and then infused back to the donor simultaneously. 2 viruses can be mixed together. A vector could be created to generate a bicistronic approach that encodes 2 separate chimeric antigen receptors so that every T-cell will express both CARs, every T-cell gets transduced. And there's this idea of a single stalk or tandem CAR approach where you can put 2 antibody binders on a single chimeric antigen receptor protein. And so the 2 virus approach has actually been investigated clinically, and this is some really interesting results that were just presented at this past ASH that I would share that with you as further introduction. So they mix 2 viruses together and end up with 3 populations of CAR T cells. Some CAR T cells that take up both viruses, some that take up one or the other. And they show that here is from the group at Seattle Children's with the 3 populations. This is in pediatric ALL, where they're using a CD19 and CD22 chimeric antigen receptors. And the really interesting thing to me was that despite the fact that at pre-infusion, the product dispersed column, the cell ratios between the different transduction patterns were quite equal. Once they were infused into the patient, really you could see in this first iteration of the trial, the CD19 CAR product really dominated the expansion and persistence, possibly mitigating the benefit of a dual CAR approach. So they noted that as well and went back to the drawing board, and they swapped out their CD22 CAR for a different CD22 CAR, one that had been reported to be highly active by the group at the NCI. And then repeated the study in another cohort of patients and found that in this case, the CD22 CAR really out-expanded and out-persisted the CD19 CAR. So I thought this was a fascinating trial, really strong work with the Seattle Children's Group, but it does highlight some of the challenges of having separate cells to deliver the dual CAR benefit. So Carlos in my lab at the time, he's now gone on to run his own group in his hometown of Barcelona. He investigated the CAR-pooled approach and compared that to a bicistronic approach and then to a single stalk approach. And I won't have time to go through all this data. It's been published recently, but just to highlight the results we found in different mouse models using either a mixed tumor that included a BCMA knockout or with wild-type tumor, there were subtly different results. So when there was the presence of BCMA knockout cells, he found that the bicistronic did just as well as the CAR-pooled approach, but these were both superior to the single-stalk approach in our hands. And the single-stalk approach, you can imagine, is challenging. Others have looked into and published on this as well. Challenging to really get optimal targeting of both antigens, given that there's a membrane proximal and membrane distal binder, which may be optimal or suboptimal, depending on the situation. And then when he looked at mice that exclusively had antigen, tumor cells with both antigens, he found there was a clear difference between the bicistronic approach versus the tooled approach, and went on to investigate the mechanism of that difference and found that there may be increased avidity when both CARs are on the T cell and both targets are present in the target cell. And so we're excited to be moving forward a dual targeted trial shortly. That's, of course, in hematologic malignancies where we're more concerned about preventing relapse and preventing antigen escape than we are about safety. In solid tumors, there is, of course, we need to improve the efficacy, but safety with some of the targets that are expressed on normal tissues is concerned. And there are other approaches for logic gating, showing some examples here of an AND gate where the CD3 zeta activation and co-stimulation are separated so that two antigens are required for optimal activation. Not gating where there is an inhibitory signal if a second antigen that might be expressed on normal tissue is present. And then if then gating, here shown with a SIN-NASH design where the initial ligand receptor interaction does not kill but releases a transcription factor that then turns on another synthetic program to begin CAR expression, or in this other example at the end there, a CAR that becomes active only in a hypoxic microenvironment. So with that, let's spend the next few minutes really highlighting some of the challenges and directions that I think the field is going. When I sat down to make this talk and thinking about the challenges that I wanted to highlight, it was very quickly apparent that these are all being addressed in the talks that you'll hear later today. So just to put these in a little bit of perspective and get you excited for the rest of the day, in terms of next generation CARs, you'll hear a talk later from Dr. Birnbaum, who looked at the menu and saw that there are probably other options there besides CD28 and 41BB for co-stimulation. And I have a really fantastic paper up on bioRxiv right now where they've made libraries of combinations of co-stimulatory domains and have investigated this with a really nice platform to look at molecular barcodes and figure out which of these may be optimal. And this may address, as you can imagine, a number of problems, not only cytotoxicity or expansion or persistence, but also migration to solid tumors, depending on the functional assay that their group employs. There's also the issue, particularly within solid tumors, of identifying targets that might be safe. And when we limit our search to cell surface targets, we're missing a large share of the potential targets. And Dr. Chen's group has really taken on that challenge and is making chimeric antigen receptors that are specific to peptide presented in HLA. And he's published this in T cells in a recent Nature Biomedical Engineering paper, but now is also moving this forward in collaboration with Rizwan Romi and Jerry Ritz at Dana-Farber to take this into CAR and K cells. And I'm very looking forward to his talk. Of course, you're all familiar with this figure in the top right there of LNP encapsulated mRNA being injected into a patient's arm. And advances in mRNA are definitely going to be important in our gene and cell therapy field in the future. You'll hear about self-amplifying mRNA, which really opens the door for increased stability that might be important for mRNA encoding CARs or other approaches to modify the tumor microenvironment. And then from the group at Tevard Biosciences, how they can use tRNA to address certain genetic diseases that encode premature stop codons. Of course, mRNA is not only a therapeutic, but it is also a target for therapeutics. And we're very lucky to have Dr. Zhang with us, who has been identifying new CRISPRs and editing RNA with them already since 2017. And he continues to discover new CRISPR-Cas proteins, in this case, very short proteins that accomplish the goal, which is incorporated into an AAV vector for mRNA engineering. And then manufacturing, I think manufacturing is a huge part of what we do. There are some really exciting talks on advances in manufacturing, including the identification of really optimal AAV production mechanisms, ways to analyze the attributes of our CAR T cell or other cell therapy products, the use of a single-stranded DNA vector for gene insertions for CRISPR-based gene knock-in engineering. And I'm very happy to be working with Polaris and Kano in our lab using some of their reagents, quite promising. The group from Kitapen, which is looking at next generation technologies to get your material, be it in RNP or mRNA, into cells for clinical translation to really enhance the viability and efficacy of those approaches. And then ways to scale up. As more and more diseases are addressed by cell therapy, there'll be larger need, and these two groups here are thinking about how to really use automation to increase scale. And a great talk from Ron Zhang, I'm sure, highlighting the new landmark bio facility that serves a lot of the Boston academic community, or will in the future, in terms of manufacturing cell, gene, mRNA therapies for patients. And then lastly, just thinking about the big picture in terms of manufacturing. A lot of what I spoke about today is in regards to autologous CAR T cell therapies, but of course the future holds several different avenues. There's allogeneic off-the-shelf approach, which can be achieved through several mechanisms, including induced pluripotent stem cell derived NK cells and T cells. But that same technology, induced pluripotent stem cells, can be made to differentiate into other cells. And you'll hear great talk about using those for hepatocyte organoid development and the implications that can bring. And then ultimately, I think the goal for many of us is to have in situ manufacturing, where CRISPR-based or mRNA-based therapies can be delivered either to the liver or even locally to lymphocytes. And this is just an example here that was recently published of doing exactly that, delivering CRISPR and gene-modifying cells in the liver. I think we'll see a lot more of that, and that'll be relevant to several of the talks today. So in summary, cellular and genetic-based immunotherapies are rapidly translational with the potential for substantial efficacy even in the most heavily pretreated patients, and I hope I was able to demonstrate an example of that with our GPRC5D CAR T-cell work. I believe advances in design and bioengineering and manufacturing will drive improvement in patient outcomes. And given the rapid advances in technology and practical advantages of cell, gene, and mRNA clinical translation, these platforms are likely to supplant traditional recombinant biologics over the next several years as the go-to for our therapies. So this is an exciting time. We had a great first inning, as John mentioned. I'm super grateful and excited to be doing this in Boston, where there's really no other city like it for scientific innovation, and I'm very excited to discover what the next 8 innings look like with the folks in this room. So I just want to acknowledge my current team in the lab at Dana-Farber, as well as Meta and Carlos, who did a lot of the work that I presented to you today, Sham Melankodi and Omar Nadeem, who are running the clinical trials and funding sources. And thank you, and I have my email address up there at the beginning. At the end, very sad not to be there in Cambridge with you, but please do reach out. And then just a shameless plug, we're always recruiting, but we have a particularly exciting opportunity for immunotherapy antibody engineering screening program at Dana-Farber that we're opening. So if you have a friend, please reach out. Thank you very much.
Thank you, Eric. That was really interesting. Please feel free to put your questions in Pigeonhole. And we do have one question. If you were to add a third arm, or maybe more, in addition to what the 2 arms that you've already done, what might those be, and what therapies would they include?
I assume the question means what more can we do besides dual targeting? Of course, there is the option of adding a third or a fourth target, but I think that eventually you can benefit even more from doing other things that might influence the way that the cells interact with the tumor microenvironment. And there are many groups that are looking at getting CAR T cells to behave as micropharmacies, which to secrete cytokines, to secrete other antibody molecules such as checkpoint blockade molecules. CD40 ligand is a particularly exciting target that can be expressed on the T cell, and folks have looked at that. So I think there's so much that can be done with these cells to really increase their efficacy in a suppressive tumor microenvironment, be that the extramedullary disease from myeloma or solid tumors.
Great. And I have a question. How different would that look if you did it for solid tumors as opposed to a blood cell cancer?
Yeah, there are a lot of us that think that's going to be super important, of course, and are trying to see the same results, the same really dramatic results I highlighted at the beginning of the talk that we're seeing in blood cancers to solid tumors, and we're not quite there yet. I think the key challenges in solid tumors, which I alluded to, are one, having the right target or combination of targets, and the added challenge there for blood cancers, it's quite easy. We can go after lineage markers, and particularly on the B cell side, the plasma cell side, it's okay if we eliminate all of those normal B and plasma cells, at least for a while. For solid tumors, of course, that's not the case. We can't eliminate all of the normal colon epithelium or what have you. So the target selection becomes much more of a challenge, and is a balance between getting cells, getting targets that are expressed on 100% of the malignant cells or near that, versus ones that might be expressed on the normal tissue. So that's one of the challenges. And the other challenge is the more suppressive microenvironment, which really excludes T cells. And there are lots of combination therapies or other approaches that people are trying. And I do think that we will start to see advances in solid tumors over the next few years.
Another question. Are in vivo preclinical animal studies a prerequisite to enter the clinic with these exciting novel treatments?
I think they are. I would be hard pressed to really anoint a candidate as a lead candidate if I didn't see some efficacy and at least some minimal safety in animal studies. I'm happy to be proven wrong on that. There are great advances with organoids and other potential in vivo sparing models, but I know the data that we've obtained both from the efficacy and the safety side in vivo models has been super important in terms of both our confidence in expending all those resources to bring something to the clinic, and also for the FDA. Now, if that has to be non-human primates, that's another question altogether, and I think probably the answer there is not always. Yeah, again, I'd be happy to talk more about that.
And we can do one more question. What do you expect to happen with the cost of developing these cell and gene therapies as we go forward?
Thank you. No, I think obviously the financial toxicity is something that needs to be addressed in this field, particularly if you think about going from one cell therapy and then relapsing and getting another cell therapy. It's one thing if these therapies are really curing the vast majority of patients, which is the goal. But so in addition to, I think, the efficacy improving and really providing more value, as the field moves towards more third-party solutions, such as off-the-shelf or in situ delivery, then certainly the manufacturing costs will come down, and we'll see. But hopefully that will result in some decreased financial toxicity therapies that become more and more mainstream.
Great. Thank you so much, Eric.
Thank you.